Welcome back to From Code to Cure, Ainnocence webinar series, where we unpack how AI is changing the way we do biology. Today's episode is multimodal biology, the new map from sequence to cell state. I'm Kevin, and with me is Dr. Pan, founder and CEO of Ainnocence.
Great to be here. Multimodal modeling is where biology complexity finally meet AI at the right scale.
So this session is pre-recorded, but we're watching the comments. If you see something that sparks a thought, drop it below, and we'd love to hear your perspective. And without further ado, let's talk more about multimodal biology. So when we say multimodal biology, what are we actually talking about?
Yeah, good question. So as you guys know that biology is a very complex system. It's from very microscopic level from molecule that consists of the biology system, like all the DNA, RNA, protein. And also we can also observe biology from a macroscopic level that we can see the cell image, and also we could observe all its bioactivities through different measurements, the different type of signals from fluorescence and to different type of functional assays that we call the phenotype that appears in the cellular tissue, organ, or even the entire body level. So as you can see that there are so many components of biology that the measurable components from a microscopic scale to a large microscopic scale, that they are all a data that can be accumulated and to tell something about the system. So we wanted to put them all together and also make sense of their relationship to each other, so that we call the multimodal biology would be observing at different scales and then put all the information together to interpret the biologic functions as a whole.
So you mentioned different data types or data domains such as DNA, RNA, proteins, imaging, and others. And when we talk about multimodal biology, we're connecting them together. And so how is this different from traditional one data set at a time analysis where we only analyze protein or only analyze DNA?
Yeah, definitely. Since in the past, the one data set mostly means the data set that generates from one type of measurement or at one molecular precision level. But and now like multimodal would be first of all, it's multi-data set. Those data would be from different measurement at different time, and it could be the same time, but from different instruments at different precision level. So in that case, the main difference is we combine data from different perspective and different precision level all together for one biological system and try to interpret and learn what is going on with that system.
Right. And so in terms of where multimodal models are right now, where are multimodal models already showing value in where we stand today?
I think there's also a long question that puzzles the scientific field is how to make sense between the relationship of genome type and phenotype, which means how we connect the understanding at the molecular biology level, the sequence of our DNA, RNA, and protein, and to relate it to like a whole level of phenotype, either a cell death or life or stress level or like an organ level. And the genotype has completely different descriptions of knowledge, and it's really hard to make sense between the two layers. But the multimodal model would give us a way to put them all together and approximate it to the same label, such as healthy and disease stage, and make sure they have some inherent internal connections to be discovered. So I think that's the first value. The second value is in the past for we call the one data set at a time type of measurement analysis, we normally would require a very large sample size to be able to interpret a certain question we try posted to that data set. But if we incorporate like a multimodal model, we could actually interpret even small sample. What is the biologic system at work inside? We're basically adding more features to describe that system and be able to even have less sample, we'll be able to still get the result we want. So there is two benefit for this type of models. Yeah.
You mentioned a lot of the benefits to these models. Currently, where we stand today, are there any tangible wins or are there any feasible uses for these multimodal models?
So, for example, if we have one blood draw from a patient, we could measure like the normal blood work for different enzyme expression, some also the minerals and different type of regular blood work, biochemical measurement. But at the same time, we can also utilize that for different DNA, transcriptome, and protein expression tests. And at the same time, we could exhaust it all the information in that one tube of blood and generate multiple data sets. And that data sets would tell us a lot more for these patients than a single measurement. Just for example, just if you just measure blood sugar level, you only tell that part of the health of this patient. So I think to leverage multimodal data from any like biological sample we can acquire, it definitely is the best use of that sample and be able to have more evidence and more knowledge and less like biological wastes.
Yeah, definitely. I can imagine how one blood draw you can get multiple layers of data and stronger evidence with fewer wasted assays or fewer wasted procedures. So that's definitely a win for where we are right now. And so, in terms of multimodal biology, which parts of discovery could multimodal AI disrupt the most right now or in the future?
We already actually see the disruption in at least two aspects. One is disease diagnostics. So in the past, we diagnosed from doctors' observations and questioning and some image and some other clinical measurement. As a standard practice now for a patient, before we reach a diagnose, we will do many level of measurements from multi-omics to image to all the other measurement we can, and we would combine all of them and make a more informed decision on the diagnose. So I think right now we're already in clinical practice, multimodal diagnostic. It achieve like a more correct diagnose rate than the conventional way. So that's one thing. And another thing is to understand disease biology better, particularly for target identification. Researchers has been put multi-omic data along with image and all the other clinical records and the type of medical history and drug has been taken, all that information together and be able to find the knowledge between those processes and variables, and then they be able to identify the root cause of the disease from a molecular level, such as certain genetic mutation or a certain abnormality of the protein expression that cause the final phenotypes. Those are also contribute greatly for developing new therapies in the next stage. So I would definitely say that there's a huge disruptive trend in medical science that multimodal AI is very useful in many levels.
So we all know the million-dollar question. So in terms of using multiomics or other multimodal biology data, there's a trade-off between speed or accuracy. For this type of data, what do we need to look for in terms of speed and or accuracy?
I would say accuracy definitely improve a lot more because we have more data and more relevant information. And then for the speed, it's really case by case. It definitely costs more time to make more level of measurement and collect all the data from different levels. But at the same time, there's also improved technology that there's certain device that could measure multiple dimension in a biologic system at once and in a much quicker turnaround time. And there's different improvement in that aspect that speed up the original measurement cost. So I would say like in the end, accuracy is what we want and speed, the faster the better. But I think speed is not what we're struggling about right now because currently I think the speed is fine. Yeah.
So in terms of accuracy, since this is definitely a high impact area, what type of accuracy can be considered clinically significant or biologically significant for target discovery or clinical diagnostics in terms of using this data?
Yeah, so that's a good question. Of course, ideally, if we have a pre-trained cross model, like could be generalized in different scenario in the future, that is great. I would say, for example, systems such as ChatGPT, they already accumulate a lot of knowledge-based data and in different levels, which some which is also a multimodal model itself. That could do some work. But in terms of accuracy, there's also other medical science specific large language model is also trying to integrate all those knowledge specifically for medical science with more of high data quality and also a better algorithm for medical diagnostics and disease biology research. So I would say I think pre-trained course model is actually a good idea, and also for institutions to have resource, they are all working on it. So I think it's probably the future trend. But currently, I would say normally in different disease area, they would have a more of a specialized model for that particular type of diagnostic or disease biology, such as, for example, breast cancer. It would be accumulate all the breast cancer image and also the biomarkers associated with breast cancer and all the other information that could have a very high precision diagnostics and treatment plan. But that data is useless for, let's say, pancreatic cancer or lung cancer patients. So there's a balance between a single model and also more of a specialized model.
Right. And so, yeah, you've mentioned the pre-trained models. And so I want to move on to the next area about hype versus reality. Right now, so all of this cross-modal models and domain-specific models, there's a lot of hype around them. So what's overhyped and what's actually real and new about multimodal biology?
There's definitely hype about one model for all biology, but the real challenge is how do you define it to be included in that one model and for what specific scientific question? Let's say if we say one model for a particular cell type, and that's probably doable, that we collect all the multiomic information of that particular cell type, and we also measure its image and its life cycle, and we probably can model it really accurately. But if we would like to see one model for a person, that would be pretty hard, much harder than at the cellular level. I think it's still at a hype region people are talking about, such as virtual human. But in terms of virtual cell, I think it's more close to what people expected it to behave. I think the main important is when we try to answer a biologic system question, we need to define clearly for that system how much data is enough and what's the data quality, and then what's the potential gap in the theory that we overinterpret the data or there's bias in the data. So we need to know all that. I would say there's some hype there, but there's also some real achievement already made in the field.
So now let's talk about Innocence's approach. So how is Innocence approaching multimodal biology?
Yeah, good question. We have been using multi-omic data for target identification, and in those data, we're also including patients' medical history and also different type of genomic like markers that at the DNA, RNA, and protein level, and to be able to help certain client to solve the precision medicine problem. And at the same time, we also leverage in terms of drug design, we also leverage data from different level of pharmacology that we from in vitro to in vivo to clinical study. We're trying to integrate all those data to make a comprehensive judgment on how a molecule would be success clinically in the end. So in terms of both target ID and drug design, we are more and more shift from a single data set analysis to a more of a multimodal interpretation.
So yeah, I know since this approach is definitely following more and more of the multimodal biology, and it's closer to the reality compared to the actual hype behind multimodal biology. And Dr. Pan, do you have any final thoughts regarding multimodal biology?
I think in the future, we could imagine a future that a patient go to a clinic and get scanned with a minimum, like taking from him, like blood or tissue, whatever, as less hazard as possible to get his medical information. And then the AI would quickly assemble all the data he get from the patient, from physiological to other phenotypes, and then it quickly analyze the results of what the health conditions of this patient and what are the targets that could be intervened to reverse these health conditions to a better state. And then the AI would also assume to analyze all the possible treatment, chemically or physically, and give him a tailored solution from prescription to physical therapies to nutrition plans, everything. And then in the end, and then when he worked out the clinic, he has the one solution just tailored for his or her self. I think that would be the future of how we actually leverage multimodal biology to solve real medical needs. And I think that future is not too far away.
I can definitely imagine considering all of the multimodal biology and all of the AI surrounding and data surrounding multimodal biology that this definitely can be built in the near future. And so thank you everybody for joining and drop questions in the comments. We're always listening. Thank you.
Yeah, thank you very much for your attendance. Thank you.